Bertrand Thirion, who is a researcher, a director of research at INRIA in France, very much involved in neurospin, etc. And Bertrand developed statistical methods and machine learning for functional brain imaging. Certainly, you may know him also because he's initiated scikit-learn, and quite famous for that. Bertrand has led several institutions. He played several roles on the plateau of Paris-Saclay. For instance, he was the leader of Data AI, our AI center at the University of Paris-Saclay. I think he has done that for 4 years. He was also the scientific head of INRIA Saclay. And very recently, he got elected as a member of Académie des Sciences. And now, Bertrand, you have the floor.
Thanks a lot for the invitation and for the introduction. So this morning, I'm going to share you some views on how AI can empower, improve brain imaging. I'm going to make 2 disclaimers. First, I won't cover all the topics. So in neuroscience, there are many different topics. For instance, one of them would be how to better reconstruct brain images, or to better acquire brain images with deep learning techniques, compressed sensing IDs, etc. And this I won't cover. Or brain-computer interfaces, which is a super interesting topic. So I won't have time to go into all that. But if you want to discuss that, of course, it will be possible later. I will be also very light on formalism. I rather want to give you an overview of what I think are interesting questions that can be addressed with AI technology. So I give you an overview, and again, if you want to discuss, and we can go more into formalism later, I can give you pointers, etc. All right. So the understanding of the brain, as you know, is very complex because you have 1 billion neurons, and that the brain is an integrated organ. You cannot simply take a small portion of the brain and look at it, look at the activity there, and make sense of it, because the brain is integrated with long-range connections. So you need to observe it globally if you want to make a decent idea of how it works. Okay, so you have to address the multi-scale problem from the beginning. And we actually have currently no good modality to observe the brain at all the relevant scales, and it would be actually extremely difficult and challenging to model the 1 billion neurons interacting, exchanging information through synapses, etc. So people have thought maybe we could simulate it if we cannot observe it. This was the idea of the Human Brain Project, which did not go very far because, of course, there are too many unknowns. We don't have enough constraints provided by the current experimental data to be able to build something like a brain at the moment. So we will take a much more humble and realistic point of view, and we will start where we can start, where we have data, where we have information. And I think that what the community has built so far is some knowledge, and this knowledge has been aggregated in publications and in public data. And so what I'm going to explain first is how do we exploit, do we leverage these resources to make good models of the brain. Maybe a slight point here. Of course, there are publications. Publications are noisy. Okay, they give some information which is not always reliable. For instance, there are some ongoing debates in the community, like if we study a language in the brain, can we say whether there are some subsystems dedicated to semantics versus some others dedicated to syntax? And you can find both theses occurring even in the very recent literature. Some people say everything is everywhere, and some people say, no, there are different modules doing syntax or semantics, okay? And so the literature is blurry because people come with their hypotheses, they test them, and they somehow build a story from the data they have and their prior hypothesis that that will say adopt one thesis or the opposite. But yeah, this posterior distribution of quasi-validated hypothesis is super noisy. Okay. Maybe one of the core issues behind that is that neuroscientists have been super ambitious. They've been asking super complex questions with limited data. And when you do that, at some point, you end up with contradictions and inconsistent views. And so what data science can do then is to help build a consensus from the existing data. So for that, we have public resources like image databases. So NeuroVault is one of them. It's a repository where people can publish, when they publish an article based on functional magnetic resonance imaging, they can provide their data together with annotation. So functional magnetic resonance imaging, fMRI, is one of the main modalities to explore brain function. So it measures metabolic activity in the brain, and so it is used to localize brain function. So you have people doing a given task, and then you compare their activity, brain activity, when they do a task, versus when they do a control task, and then you obtain the correlates of the task you're interested in. You can also upload your raw data on Open Neuro so that people can reanalyze them. People have also provided or started to provide a knowledge base for cognitive functions. We have currently no atlas of cognitive functions, no model of actually how our mind works. Okay, so we are still trying to figure out how this works. So people just try to enumerate possible concepts that will be relevant, but we don't have a rigorous structure on these concepts. Recently, so Europe has developed its own GDPR-compliant neuroimaging data repository, eBrains. Okay, so with that, what can AI do? Well, AI is good at building foundation models, so we're going to see how this has occurred in the field of brain imaging. It is good at bridging between heterogeneous data sources, and so we will show how we can put together images, tables, and text to build a better model out of all these data. And then in the second part of the presentation, I will also show how the cognitive models provided by AI on language, on vision in particular, can be used also to better understand the brain. Okay, so let's start with building a foundation model of the brain. This was done 10 years ago. We would not call that a foundation model at the time. So by a PhD student, Arthur Mensch, who is now is now the CEO of Mistral AI. He's a billionaire. So this is the destiny of a PhD student. So what we did at that time with Arthur was to take all the existing data. So these things are brain maps, so they cover the brain with a millimeter resolution. You see in color-coded the strengths of brain activity in response to a given task. You also have labels that describe what the subject was doing while these data were acquired. And so you've got that for many different data sets. You also have fMRI data without labels, so data acquired during rest. So quite a lot of data, terabytes of data. And what we did with that was a shallow neural network that would first do a compression of the data to obtain a more manageable representation. And then the output of the network would be to be able to classify for each of the input data sets what experiment was, or what condition was present. So for instance, you could have a horizontal checkerboard versus a vertical checkerboard, and you would like to be able to distinguish between images corresponding to one or the other. And so you've got a classification head for each of the different data sets, but very importantly, there is a bottleneck layer that mixes information between all the different classification tasks so that you build a common representation of the cognition across all these data sets. So another way to see that is that we create a suitable brain representation that can capture the experimental conditions across a vast array of tasks. Okay, so when we do that, there are about 40 tasks used at the time. We can look at the decoding accuracy. So the small red bars here indicate the chance level. So we do better than chance in all cases, essentially. But the important thing is that this multi-study decoder that mixes data from all the 40 studies together obtains, in black, a higher accuracy than the decoder that would just use the data from each study in isolation. Okay, and we can map the difference on balanced accuracy here on the right side, and there is an average again of 5%, and we obtain again in 80% of the studies. So there is a transfer learning occurring through this bottleneck layer of the predictive network. More importantly, we can look at the intermediate representation, so in the bottleneck layer actually, and we can try and disentangle the different components of interest. So the components, it's essentially a dictionary learning approach where we describe this latent space by finding a relevant basis to describe it. So the basis is made of topographies, so sets of brain regions, like that one, that correspond also, and you can associate to it a word cloud that represents the cognitive conditions that were well discriminated by this network. In that case, it would be a motor task corresponding, because it's the region here of the right hand, and so you can see that it's good at isolating conditions involving right hand movement. But you've got that for different systems, auditory, face responsive regions, etc. So as reviewer said, we did not discover much because we already knew many of this mapping, but here, for the first time, they are obtained from a consensus of all the public data available. And this is available. Okay. Then we pushed the game a bit further. Now you will give me a brain scan of the experiment you did this morning, and I will try to guess what the subject was doing. This is a more open-ended problem because there might be multiple relevant labels to describe what was occurring during this experiment. And so it's no longer a multi-class problem, a multi-label one. And so with lots of possibilities in terms of conditions. So to do that in practice, we use a NeuroVault database that I talked about, in which we could select about 50,000 maps that were relevant. So these are brain maps like this thing, so brain volumes, again with this activity showing the regions that were activity during a given task. With this data, we just pre-process them to obtain a kind of normalization, standardization of the signal, and we compress them to build a manageable representation which is 1,000 dimensional, much smaller than the input image. And then comes the difficult part, which is to associate labels with this data, because we only had user-supplied labels, but they were extremely noisy. And so we had to really decode or guess what was the intent of this experiment and what was mapped with the image. So we extracted the concept. We tried to align those to a given cognitive atlas, which is a work-in-progress ontology of brain processes. And so we were able to associate labels to this brain data and then to do the decoding. So what we did, so we call this NNOD, which is Neural Networks on Dictionary. We can look at the accuracy of the prediction of different cognitive terms that we were able to test. There are about 50 of them. We obtained essentially better than chance AUC in essentially all the cases. We did better than previous approaches, but previous approaches were not built for decoding. They were built for mapping, and decoding is a bit different, so it's not surprising that we did better at decoding. And we obtain relevant topographies for many concepts, and sometimes it failed on a few of them. So just another view of the same results. The AUC for the classification accuracy, so we do essentially better than chance for about 50 concepts. So we can look a bit more at what happened, open the box, essentially to understand the decoding rule. Some cases are trivial and we can perfectly decode what happened, like when people have to move their left toe or right toe in the scanner, there are some brain territories dedicated to that and there is no possible mistake on it, so we can perfectly decode that. Now, more interesting, we can study cases where tasks involving syntax processing, like having a complex sentence with a relative, etc., versus a simple list of words or very simple sentence. And so we can study the impact of syntax on brain activity to map syntax. If we just look at the correlates of syntax, we would obtain all the language network, which is this reddish area here. But now if we try to decode, it's a bit different because we have a discriminative approach and we look at the regions that bring information on top of the just standard semantic or language processing. And so we obtain a finer decoding where here the anterior part of the supratemporal sulcus or the inferior frontal gyrus are implied. And this is probably a better map of syntax processing than what we obtain by just looking at the correlates. And there are some failures. Example of a failure is when you try to decode face perception, so the brain activity when people perceive a face in an image. Well, you've obtained the fusiform face area, which is fine. Then if you do a face recognition task, what you obtain is the same thing, plus a little bit of left motor activity corresponding to the right hand, which means that when people recognize a face, they just see the face and press a button, and this is all you map. So there is no such thing as a face recognition process. It's just the addition of two trivial things. But even worse, when you study emotion perception, you obtain, again, the face areas, because in cognitive neuroscience, when you want to elicit negative emotions, you use like fearful or weird face images. And this is the easiest way to elicit emotions. So at the end, the classifier for emotions is completely confounded by this effect. And so this just reveals the experimental biases of our data. Okay, so what we discovered by doing this is that the difficult part, the most difficult one, was to really map the concepts. As always in machine learning, okay, what is expensive are the labels, not the data. So how to get better labels? So we could use the literature, which provides structured, meaningful text that describes cognitive processes. And so we started to look at the neuroscience and cognitive literature. The idea will be to leverage the text from which we can extract topics, concepts, etc., and then study how they can be encoded to brain activity, or whether we can decode brain activity. We can also build brain representation from the data, and etc. So this is this. So we do modern data science with neuroimaging data, and indeed, standard sentences in the neuroscience literature are very informative about the organization of brain regions and their relationship to cognitive processes. So what we did, so essentially when you take a text from the brain imaging literature, what you have is the text itself that you can extract. And then what we did, it was 7, 8 years ago, so we did not have good LLMs at the time. So we did just a bag-of-word approach by taking the TF-IDF of the text, which would just reveal the concepts that occur more in the given text than in average in the literature. And though it's a way to summarize, to have a sparse summary of the text by just taking the concepts that pop up in the text somehow. And this will be the X feature for each given text, and we can organize these features in a matrix, where the rows are the different text and the columns are the different concepts. So we have an occurrence matrix here, the occurrence of concepts in a text. In the articles, you also have like this kind of information, which are tables providing coordinates in a three-dimensional brain space. And if we just put a kernel on each of these locations, we obtain a brain map like that one that essentially represents the elicited activation during the experiment. And what we're going to do is to try to relate statistically the word concept occurrence to the resulting brain image. And for that, we will find regression coefficients. And the good thing with that is that then you can, since it's predictive, you can try to predict a new y for a given x. And so you can really measure how well this generalizes. One of the twists is that for you have this 1 over f fate, which is that very few concepts are mapped a lot across the literature, and most of them are mapped very rarely. So you would not have enough data points to map them reliably. So what we found was the idea of doing a query expansion. So if you take the term melody, there is not a huge literature that relates melody to brain data, but you can identify that melody is semantically related to music, musical, auditory, and pitch. And many of these terms are frequent enough that you can map them. And so at the end, you can propose a map corresponding to melody. When you do so, you deliberately bias the user query from melody, which was the initial query, to something that you provide, which is not exactly melody. But here you have the traditional bias-variance trade-off, where you trade a bit of bias to reduce the variance. Okay? Since you have much more data to map it, you have a low variance, and at the end of the day, your error is smaller. So and we check that with cross-validation, that typically you better bias the query to obtain a result with lower variance. And yeah, in average, at least it will work better. And we did some validation also on external data, like brain image repositories, checking that the output of our system was close enough, for instance, to what you would obtain on real data for a given concept here. Read pseudo words versus read consonant strings. And so we built the neuroquery interface, and you can put any terms. It will analyze the terms in your query, try to expand it across a vocabulary of terms which are well mapped to the brain, and then propose a brain map that summarizes what could be the reasonable correlates of your query. Okay, so now this was a kind of state of the art 6 years ago. So let's try to do a bit better. First thing, we built the brain images with the coordinates by putting kernels. Maybe we can do a bit better. For instance, we can take a database of images, extract the peak of these images, and given this information, try to reconstruct the image or a vectorial representation of the image. Okay? And so for that, we can use, for instance, set transformers to do this reconstruction, which might be more accurate than just putting a kernel, which is a bit minimal. So we did that, and we actually observed that in terms of reconstruction loss, this approach of the set transformer would obtain a better reconstruction of the images than using kernel-based techniques, for instance. Then, once you have created this set transformer, you can take all the neuroscientific publications that you have in the literature, throw the coordinates reported into the set transformer to obtain a brain image. Then, in parallel, you extract the text from the publication. You obtain the well-represented concept in the document. And then you can play the game of learning the association between the concepts and the brain maps. And for instance, you can do decoding by training a classifier to predict a given term given the topography described in the article and reconstructed thanks to the set transformer. So when you do that, you obtain actually for different terms. So for like here there were more than 100 terms. Here are just a few of them. We could obtain a topography for them, which is, I would say, relatively reasonable. Not very detailed, but overall it gives decent ideas of the main brain network and their association with different cognitive terms. But of course, we want to validate this kind of finding, and for that, we take this classifier that has been trained on the literature only, okay, and we throw real data into this classifier to see if we can recover the labels that we think should be associated with each given sample in the image database. And so we could do that for more than 100 concepts, and we obtained better than chance accuracy in maybe 80% of the cases. It's the first time we are able to decode at scale more than 100 concepts. So we do particularly well for language or executive cognitive control, which are pretty sophisticated tasks. Surprisingly, we do quite poorly for like perception terms. Maybe these concepts that are occurring a lot in the literature have more variability, are used more inconsistently, and this is why we cannot map them well. But so to me, the impressive thing is that we did not do any label supervision on the images. We just trained everything on the literature, and we were able to generalize to an image database. Of course, we want to get rid of this old-fashioned TF-IDF approach to extract the concepts from the text, so we can now use LLM embeddings for that. So you take a pre-trained language model, and then you create an embedding of the text of the publication that you can compare to an embedding of the, for instance, the locations reported in the article that you represent with your set transformer or your kernel estimate. And then using a contrastive loss, you can learn to associate the text embeddings with the coordinates embeddings. And so to measure how it works, we can do things like retrieving text given the terms in text. Can we retrieve the locations reported, etc. So these kind of things. And we observed that essentially our approach was much stronger than state-of-the-art to obtain good retrieval. So in terms of Recola 10, we obtained 25%, which is not that impressive yet, but it's much better than previous attempts. And we could also do some ablation analysis. At the moment, well, when we did that 2-3 years ago, Mistral 7B was performing a bit better, for instance, than alternatives in terms of LLM. But of course, this might change in the future when we have more data and better LLMs, obviously. We can also use the system also to decode the latent representation and be able to reconstruct an image. And again, we can, so this is based only on the literature, all that, so we can reconstruct the correlates of some concepts in cognitive neuroscience, like reading a Jabberwocky, which are sentences with a correct syntactic structure but scrambled semantics, versus reading pseudo-words. So to see how the brain encodes syntactic information. And we see that with this approach, we obtain results well, reasonably close to what we obtain on actual data. Okay, so at that point, I think that the next frontiers are to do that on more medical applications. Here I was confined to cognitive imaging, but of course we would like to be able to map diseases in psychiatry and neurology. One of the limitations is that there are fewer available data because it's complicated to share medical data or data about mental health. And also there is a cultural challenge that this community maybe is less used to sharing data. Another point, technical point that I'm not going to discuss because it would take me half an hour, is the question of interpretability. When I show you images showing regions in red that are predictive of something, well, how do we make the inference that this particular feature or region is predictive about the outcome? In practice, when you want to do that, you do a conditional independence test, which is pretty challenging in high dimension. But so there have been a lot of progress in the literature in the last 10 years on this question, with many works of Candace and also many works on, of our group on this. So I'm happy to discuss that more in detail. But so to conclude on this first part about data science for brain imaging, so the idea is really to extract weak signals from large amounts of data, here the literature, use bottleneck layers which create common representations across data sets which make stronger representations. Again, the hard part is to identify commonalities across different studies, but AI brings heteromodal learning, and of course LLM are a great tool for that. So, but all this rests on public resources, so the public data sets that I mentioned, and it's really important that they keep growing with involvement of the community. Okay, I jump to my second part, which is maybe a bit more original, which is that since 12, maybe 15 years now, we've had AI-supplied cognitive models, and this provides suggestive model for how the brain could solve some problems. And so, how can we leverage that? Maybe I'm going to jump a few slides because time is going fast. Maybe that one. Oops. Did something wrong. For instance, we may want to map what happens in the brain when the brain is, when the person, is looking at an image. Okay, so we know how to map a given image, which is defined in a pixel space, to some relevant feature space, so we can embed images with your preferred computer vision embedder, and then we can map these feature level representations to the brain space, so we can study how the features extracted from the image predict activity in each brain region or in each brain location. If we can do linear mapping going from features to voxels, we can also do the converse mapping, which consists in identifying the features of the image given the voxel activity, so the activity in the brain region. And since now a few years, we've been able to go all the way back using generative models to rebuild images from the features that have been reconstructed. So a few years ago, a big data set became available, the natural scenes data set, where people viewed up to 9,000 different images. And this is good data acquired on a very high-field MRI scanner. And so people started to reconstruct the images perceived by the participant in relatively complex scenes. So here is how it works. So essentially at training stage, you look at the brain activity while the subject is looking at an image like that one. With your preferred vision model, you can encode a representation of this stimulus, and you also have a vector of activity regarding that represents brain activity, and then you can learn to predict the image embeddings from the brain data by regression, essentially. Then at test time, the subject uses an image, you extract the brain activity. With your trained regressor, you can estimate the latence of the presented image, and then with this estimated latence, try to reconstruct the image. People often do it in two stages. I won't go too much into details, using first a relatively coarse model. We have a very deep VAE. And then they use more sophisticated diffusion models to reconstruct a more photorealistic image. And this diffusion model is then initialized by the VDVAE output and conditioned by the embedding of the estimated embedding of the image. Here they were using a clip model that encodes the image and the semantics of the image. So these predicted clip embeddings, image and text, are used to condition the reverse diffusion process to obtain a good rendering of the image. And this is really amazing. Sorry. You can, if this plain image was presented to the participant, we can indeed reconstruct from their brain activity the plain image, which captures well the layout of the scene and the semantics, obviously. And similarly here for the group of people, we see that we were not able to reconstruct exactly the image, but we were able to capture I would say the layout of the scene, that it's a kind of outdoor with people interacting, etc. You can capture some structural features of the scene, like the horizontal bar here, which is captured by the reconstruction, and so on. Okay. Of course, in this case, the reconstruction hallucinates. Okay, the rendering is very nice, but well, it may depart a bit from the actual thing, and there are some failures, obviously, which are quite terrible. But like it happens when there is something like an occlusion, so the scene is a bit hard to interpret because it's unclear whether there is a character or the body, and so the reconstruction is really poor because somehow the system was confused by the wheel structure of the scene. Sorry, it's a bit unpleasant. But yeah. So this is where we are. People are starting now to reconstruct videos watched by people just by looking at the brain activity during the video watching. What we've done recently, and we presented it just 2 months ago, was to have people listening to stories in the scanner. So we make them listen to podcasts typically, and we record brain activity while they hear the language. And so we learn to associate, again, it's the same story. We learn to associate the brain activity with the language embeddings of the podcast. And then at test time, we can try and reconstruct the sentence that was listened by the participants by predicting the language embeddings. And then you can regenerate language if we want. And it starts to work. So after 10 hours in the scanner, you start doing better than chance, and you see the scaling law somehow of this decoding. I spent last week several hours in the scanner, just scanning myself, listening to podcasts because we want to get more data and push that much further. So scaling laws. This is an interesting part. So across many different data sets and actually many different brain imaging modalities, I don't go into all details, but what you can see essentially that you have this increased decoding ability when you increase the number of trials, but you need to get to relatively large numbers to get a decent decoding. But when you have high quality data like the 7 Tesla natural scenes dataset, the curve is steeper, and so you have a better decoding. So this is within subject data. You can really improve decoding with within subject data. The catastrophe is here. When you limit the data per subject, but acquire data from more participants, then you don't gain anything because the brains are too different. There is no simple matching between brains, and so you don't generalize really better by mapping more individuals. You rather should spend your acquisition budget in more data per individual. But you won't be able to generalize then. So we want to address this question by learning to map a brain to each other. So I kept this equation. This is my equation of the talk, so I kept it. It's a model to try and align different brain data. So it's an optimal transport approach where you try to find a coupling between the brain locations from a source subject to the brain locations of a target subject. So the coupling is Pij, essentially. It's you map region I from the source to region J of the target. And so it's a non-negative number, like a probability of mapping, and you want it to be high when the functional response is similar, so the difference is low. But then you want to regularize this so that the geodesic distances in the source subject are preserved. So pairs of regions should be matched when the geodesic distance in the source space and the target space are similar enough. This is a regularization of the problem, just to avoid completely mixing the brain, which would be completely meaningless. So you want to regularize the problem. And so technically, it's a Gromov-Wasserstein term added to the loss. Then you have the terms, which is a bit horrible, but just means that you want to preserve the marginals of your coupling. So you want to map all the source locations to all target locations. And you may want to have an entropy term just for regularization and numerical convergence. The nice thing with this formulation is that it can work on different geometries, brain meshes, because you want the geodesic distances to be preserved, but actually the structure of the mesh and the shape of the mesh could be different. And so, okay, we have learned lots of experiments just to show that this would behave better than traditional diffeomorphic mapping and or procrustless alignment, but it's unmanageable, I would say, because the different terms are not homogeneous. Okay, so learning the couplings, the alphas and rhos that connect these terms is just horrible. You don't know a priori what are the right parameters, so you need to try with grid search, but this takes ages to solve with an solvers. So even with the best solvers that we have at the moment, when you use large meshes, this is just untractable. But it works, okay, when you are patient enough. I just want to say that using this alignment led us to a very interesting conclusion. I'm just skipping, sorry, the details, but the important message is here. When in a given subject, when you have limited data, then you have a very poor accuracy. So here, lower is better, okay? So this is a retrieval metric, so you should have a low value. And so when you have little data and you train a model to decode, well, this is not going to work well. You should rather align the subjects to a population template, and this will buy you a better decoding accuracy. It's the purple line. But of course, when you increase the amount of data available, then there is a crossing at some point, and you end up having a better model, but just working on your new subject. Okay, so it's interesting to have this trade-off, and it happens with all participants that we could observe that with limited data, you rather work in the population space. So you rather want to use your data budget to map your subject to the population, and this will provide you a better decoder. But then if you have a lot of data in a given individual, then rather use it directly to decode. I just want to mention that since this fused unbalanced Gromov-Wasserstein transport was unmanageable computationally, we have recently come up with a much faster solution, so developed by Sonia Mazlet and Rémi Flammary, with graph convolutional networks that are used to approximate the fugue problem. So essentially, we learn to solve the fused unbalanced Gromov-Wasserstein loss using this relatively simple and cheap architecture, so that we make the alignment problem trivial. And so we can essentially align the brains very easily. So here we draw on the idea that a mesh is essentially a graph, and so we can use graph matching techniques like based on GCNs to map the graphs together. And this works. We can have a time reduction like of a factor 100, obtain a maybe not optimal but very decent solution, approximate solution, when using this fugue criterion. And this solution obtained with this proxy could be an excellent way to warm start, for instance, a complex solver, and so saving computation time. Okay, time to conclude. So it's super exciting now to use these modern AI representations of cognitive content, like so vision, videos, we could also play with mental imagery, sound, language. And since we do decoding of this information, I think it creates an opportunity to build new biomarkers from brain function. And this is something I'm trying to do now with a startup to improve this decoding-based, so the decoding first, and then build biomarkers from our decoding abilities. I'm also very excited to see that recently people have shown that they have built foundation models that can solve psychological tasks essentially like humans. And so now we start to have AI models, think of AI agents somehow, that solve different psychological problems similarly to humans, that obtain similar performance. And so we start to have a full chain of candidate models on which we can operate, do ablation, do lots of operation, and then compare, of course, the internal representation of these models to the brain data. So this field is moving very fast. Just last words to come back to software. So to me, it's interesting to play with data, but it's important also to deliver useful software, and that the community builds together good software. That's why it's essential to develop open source projects, open to the community. And that's what we've tried to do in the group by creating, for instance, the NILearn, which is a library that adapts the scikit-learn concepts to brain imaging. And with this, I would like to acknowledge the contribution of my colleagues, and thank you for your attention.
Thank you for the great talk. So now we have time for questions. I will, yeah, please go ahead.
So I find it very interesting that you want to use this to produce biomarkers which you could then use in medical applications. But currently we don't use fMRI very much in medical applications, and when we do, we're usually using a single resting state fMRI sequence, which doesn't tell us much about how a person's brain behaves when they're actually performing tasks. But is there some way to design we don't necessarily know what kind of behavior we want to analyze. Is there a kind of uniform fMRI task protocol which we could design to capture something like that?
No, that's a good question. So first, resting. So what we do is to have like people staying, say, 7 minutes in the scanner, just doing nothing. This is resting state. They have to keep their eyes open, not to fall asleep, and that's it. And what this is supposed to do is to kind of measure the correlation between regions. We call that integration of brain systems, but with a very coarse measure. So 7 minutes is not a lot to have reliable correlations. And so this is not very sensitive and not specific at all. It does not tell you really anything about the nature of the disease. Okay, so if we want to go beyond that, we need to use a task. We need to use an easy task, because otherwise it just won't happen. So what we believe, for instance, is that a naturalistic task, such as watching a small movie, and there are lots of even Walt Disney small movies can be great because they can elicit lots of emotions, for instance. And there is a project in France, and a psychiatrist that tries to use them to elicit emotions and to look at brain responses to those. Or listening to stories, to podcasts. This is so. As I told you, I've already spent 10 hours in the scanner listening to podcasts for 1 hour and a half, and it's super pleasant. You don't fall asleep. I mean, because for me that's a challenge. And also, I think it's a great open door to cognition. So emotional processes and cognitive processes, because you need to think of the story, who relates to what, the context, etc. And I think that these kind of naturalistic tasks are excellent for this kind of questions. But of course, this will have to be validated in the long term. Thanks.
Other questions?
Yes?
I was wondering, I mean, I think it's fascinating. I never thought this was possible, and also a little bit worrying, I think, if you can reconstruct an image of what the person is thinking about effectively. But I was wondering about the reproducibility. So you talked across the different subjects. So what about the reproducibility of the individual? So if you keep scanning the same person, let's say on the same scanner, what is the influence of the scanner as well, if you were to repeat this elsewhere, and the time of day and all these kinds of other factors? How can you control for that?
A good question. I've seen very few experiments doing that across scanners, but that's a good question. Overall, so what we see, there is pretty good consistency. The point is, we try to avoid repeating a stimulus, because the second time you see the image or the video or the story, well, it's not exactly the same for you. You may be less attentive, etc. So there would be behavioral differences. Actually, in the natural scenes dataset that I presented, they repeated the stimuli 3 times, but so the subjects were seeing almost 10,000 different images, but each of them was repeated 3 times, and actually they use to decode the average of the 3 occurrences, which probably tames some of this variability between repetitions, but it was still within scanner. So yeah, it's a good point. In practice, it's hard to organize a protocol on many different scanners. So for very pragmatic reasons, I think people have not tried that, but that would be a relevant question. My expectation is still that the subject differences between subjects are larger than the differences in between scanners. But we'll see.
Other questions? Yes.
Very fascinating for me to listen. In your slide, I think it was 30, when you went from the fMRI to basically predicting the task of the subject.
Yeah.
I find it a bit ironic to see pattern recognition as the one that do extremely bad, since you're sort of doing pattern recognition with the with your machine learning here.
Yeah.
Do you have an idea about that? Why is it so bad?
Yes, I think that well, because pattern is, I guess, a bit ill-defined.
Yeah, so that's the problem.
You can have shape recognition, you can have image recognition, various kinds of object recognition. And quite often, when you have this type of issues, is that you have mislabelings. And so what we observed, I did not insist on that, but sometimes the most frequently used terms are not well mapped because they are not used very consistently. The problem here is that on our test data sets, the labels are extremely noisy because they are provided by people, but they are essentially uncurated. So we did the curation ourselves, trying to figure out what people wanted to mean when they provide these labels. But of course, it's extremely limited. And so one of my messages to the community is please try and think of how to better labels your data, because we will need that to go to the next generation of models.
Is there any question online? Okay, so I have two short questions. First, in the approach of VDAE, where you do surrogate regression and you link with the full representation, did you analyze in your regression task, in the final regression task, for instance, did you analyze, how you can relate, some features of the input and the output? You try some interpretability somehow?
Okay. The nice story, if you want the nice story first, is that this was working super well 10 years ago when we were using convolutional networks to model vision. Because actually convolutional networks like AlexNet are very similar to what we know about the visual system. And so we did that with AlexNet. We presented AlexNet several images, and we looked at the brain activity while the subjects were looking at the same images. Sorry, it's not super nice here, but then when you look at it, so the first layer, the activation of the first layer of AlexNet would predict very well what happens here in the medial part of the visual cortex, which is V1. Then the next layers would predict V1 less well, but start to predict better what comes around V1 and these external regions. And the final layers of AlexNet would predict not V1 at all, but quite well what happens in the peripheral regions. Actually, you really have this mapping of the AlexNet layers to brain regions, which is amazing. So, of course, in the long term, you can think that since convolutional networks were derived from what we know from the cat cortex and the monkey cortex, and it's quite similar to the human cortex, actually, it's kind of, we close the loop somehow. So, now with vision transformers, with more complex architecture, the mapping is less clear. And so we haven't really tried, but indeed, I think it's still a question of, I think we need to find some intermediate, having models that retain some of the structure that we know from the brain. Otherwise, it's really hard. So people try to analyze networks with like dictionary learning to extract some components of the network that could be mapped to brain activity. So we haven't done it yet, but it's still just still in its infancy, I would say.
And second, you point out the fact that there is indeed in the domain a difficulty, and it was also during the question of how to design good experiments to really extract what you want to extract, as you said, for instance, with the perception task that is difficult to tackle. So, do you think there is a whole research topic to try to leverage experimental design, knowing that, of course, you cannot do that online with your fMRI, but
well, people are starting to think about that. For instance, it will make sense when you have a patient in psychiatry in which you may think that certain type of stimuli might be better at eliciting or revealing differences between a given individual and the standard population. And so I have a collaboration with people in Montreal that would like to do these kind of things, trying to essentially design or select some experimental material to be presented to the participant to better reveal potential differences. And so, and this, I think it would be interesting to do it online. So while we have started to scan, trying to optimize the design of the experiment to better reveal differences. I think it would be a bit brittle statistically, because how to make a proper inference based on such adaptive designs. I think it's really an open question. But when you work with very generic stimuli, such as images or speech, then just being able to select a better one or potentially generate a new stimulus that would be suitable for your participants to map a given system, I think would be a good idea. So I don't know. Maybe I would say in 10 years people will be trying these kind of things for real.
Okay. So, is there any question? Also, we have time to discuss after next week during the coffee break, and we thank you very much.
Thank you.